The pigment present in we human beings is nothing but melanin that gives us the different shades of whites and browns that are there. Females are homogametes carrying only one X chromosome. Males are heterogametes carrying an X chromosome as well as a Y chromosome. Spermatogenesis process, if you see, first the spermatogonia will undergo mitosis and then they will undergo meiosis to produce the sperm. Hello everyone, a warm welcome to this session for class 12th biology. I am Dr. Divya, biology faculty. So in this session, that is session 5 of chapter 4, principles of inheritance and variation, we will talk about polygenic inheritance, pleiotropy and sex determination in humans as well as in honeybees. So in the previous session of this chapter, we had learnt about the chromosomal theory of inheritance and also we learnt about the linkage and the recombination. So chromosomal theory of inheritance actually came into existence because of the advent of the microscope, and also people were not satisfied with whatever theories were put forward by Mendel. So Mendel's theory was the base for all the other genetic work that came up, but still there were some loopholes in that. So Mendel, what he had said was, he said that there are some factors which are present in the parents that gets carried to the progeny, wherein we can have just alternate character. Say for example, if the plant is tall and one plant is short, then the progeny will be either tall or it will be short. No intermediate characters are there. That is what he said. But there was some experiment that came up wherein it proved co-dominance, where incomplete dominance was seen, wherein both the characters get expressed, or one of the character is not completely dominating the other and an in-between character gets expressed. So all that. Or the best example that we can take of is our human skin color. Right? If that is the case, we should have either only fair people or only dark people. But we have different shades of skin color, right? Now, how can we say that Mendel's laws are completely true? No. But to some extent, it is true. But apart from that, there are some corrections to be made. So, in that, we'll talk about first thing, that is polygenic inheritance. The polygenic meaning poly means many. That is inheritance of many genes from the parents, right? Say for example, the mother is shorter, the father is taller. So either the child has to reach the height of the father or reach the height of the mother. But can it happen? No. Sometimes the child can grow taller than the father. Sometimes the child can grow in between height because we take both the genes from the mother, and that is polygenic inheritance is there. So that is about polygenic inheritance. That is when two or more genes control the traits or the character having distinct alternate forms, it is called as polygenic inheritance, meaning we inherit multiple genes from our parents. And the polygenic inheritance is also influenced by the environment. So that is a skin color. So our environment, the where we are staying also determines the skin color, because sometimes when we are exposed to too much of sun, melanin production is more. Very less sun, melanin production can be less. So all that can happen. So the best example that we can take is for polygenic inheritance is human skin color. So we'll explain about how human skin color can be taken as an example for this. So we all know that our skin color is caused because of a pigment. So pigments are the ones which offer color, right? Any be it plants, animals, whatever coloring they have, it's because of pigment. And the pigment present in we human beings is nothing but melanin that gives us the different shades of whites and browns that are there. Now the quantity of melanin is due to the presence of different genes. It can be because of genes A, B and C. Now the genotype of offspring, that is of the first generation, that is when crossing is done between the parents. So the genotype of offspring of F1 generation will have 3 dominant alleles, that is A, B and C will be there. All are dominant, and 3 recessive alleles which will give intermediate colors, that is it will have A, B, C and A, B, C. 3 dominant alleles and 3 recessive alleles will be there. Thus, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin. Now, if they inherit these 2 genes, then it will be a bit, say for example, A gene is responsible for darker skin color, C gene is responsible for lighter skin color, or small letter C is or the recessive gene is responsible for even more lighter skin color. The combination between these alleles, what the genes get, because all these, a few of these will get inherited by, so it's not that the offspring will get only the A, only B. No, either they can have a combination of these four, a combination of this and this they can receive. So based on that, the melanin production depends, and based on that, the skin color depends. So that is called as polygenic inheritance. That is, multiple genes we can inherit from our parents. Now there is one more term which is called as pleiotropy. So pleiotropy is nothing but one gene can have multiple characters. That is called as pleiotropy. Usually, many genes will have, one gene will have only one character. Okay. Now, all the genes entering into the offspring, we call it as polygenic inheritance. Now, one gene having multiple characters, we call it as pleiotropy. So, that is the difference between polygenic inheritance and pleiotropy. Now, talking about sex determination. So, this was first studied by Henking. That is, Henking studied spermatogenesis in some insects. What is spermatogenesis? The formation of the sperms, the generation of the sperms. So Henking, in the year 1891, he studied spermatogenesis in some insects, and he found out that 50% of the sperms received a nuclear structure after spermatogenesis, whereas 50% of the sperm did not receive this nuclear structure. So later on, he called that nuclear structure as X body, which was called as the X chromosome. So some of the progenies, they received the X chromosome. Some of the progenies, they did not receive the X chromosome. Later, it was observed that the ovum that receives the sperms with the X chromosome will become the female, and those which becomes the male will not receive the X chromosome. Number of autosomes is same in males and females. Sex chromosomes, that is allosomes, are X and Y. So, how many chromosomes do we have? 46, right? 23. So, in that 23 pairs of chromosome, the last two pairs that are there, one will be XX, the other will be X, and it will be Y. So, this is autosome and this is allosome. Autosome means same, allosome means different, okay. So, number of autosome is same in males and females, that is true, right. They will have 25, that is 25 autosomes both in male and the female, but in that is sex chromosome, that is allosome, X and Y are the chromosome which involve in sex determination. Say for example, now the progeny, that is during the fertilization process, if both XX chromosome goes into that progeny, it will be a female. If X and Y chromosome goes into the progeny, it will be a male. So, that is how sex is determined. So, that is why always the females are not the ones to be blamed for bearing a girl child. It is only the males who are actually responsible for the female to bear a girl child, because only if the Y chromosome enters into the gamete and it fuses with the egg and fertilization takes place, then the female progeny, that is the fetus that will be there, it will have a XY chromosome. Then only the fetus can become a girl. So, therefore, mother is not to be blamed for bearing a girl child. So, that was a very big misconception or a myth that the people had. So, that is not it. So, this is how sex determination is done in the humans. So, mechanism of sex determination, that is the male heterogamety. The male produces two different types of gametes, right? It will produce a X gamete and it will produce a Y gamete. So, these are the two different gametes the male produces, but the female produces only the X gamete. So, that when we were talking about in the previous session also, I have showed you how, right? So, that is called as the XXXY mechanism. Here, male is heterogametic. They carry X and Y chromosome, whereas the female is homogametic. They carry only the X chromosome. So, XX is female, their gamete is only X. X, Y is male and their gamete is X, and Y. Therefore, females are homogametes carrying only one X chromosome. Males are heterogametes carrying an X chromosome as well as a Y chromosome. Now, in that gamete, if X fuses, so if this X fuses with this X, then the progeny will have XX chromosome. Now, if this X fuses with the Y, then the progeny will have XY chromosome. So, therefore, this can be a female fetus, this can be a male fetus. Example, both in humans as well as in Drosophila. Next, talking about mechanism of sex determination in insects. So, in insects, what happens? There is XX-XO mechanism. So, male is heterogametic in insects. Male is heterogametic in insects means they will have a X chromosome and they will have a O chromosome. So, gametes with X and gametes without X. So, what do we call as O? O means gametes without X. And female is homogametic. All gametes are with X chromosome. That is, they will have X and X chromosome, females. Now, eggs fertilized by sperms having an X chromosome becomes a female, and those fertilized by sperms that do not have X chromosome becomes the male. Same thing here. I'll do the crossing over X and O, X and X. Now, what are the gametes here? X and O is the gamete. Here, X is the gamete. Now, when crossing over, what happens? We will get, that is in the F1 generation, we will get X. And we will get X and O. So, this will become the female, this will become the male progeny. That is how in insects that such as grasshopper and cockroach, sex determination is done. Next, mechanism of sex determination, there is one more, that is female heterogamety. So, total number of chromosomes. So far we had studied females were homogamous, that is in humans, Drosophila, grasshopper and some insects, right? Now, if females are heterogamety, what will happen? So, total number of chromosomes is same in both the male and the females. Female produce two types of gametes. What is it gametes? Male will produce ZZ, female will produce ZW. So, male will produce what they'll have? Male will have Z chromosome, homogamous. Female will have Z and W chromosome. This is heterogamous. Now, here male is homogametic, that is ZZ, and female is heterogametic, that is Z and W. Example, this happens in birds. Now, here again, what will happen? Crossing over. Okay, I'll do the crossing over. Before that, we'll write the gametes. Now, what will be the gametes? In the male, it will be Z only. In the female, it will be Z and W. Now, the F1 generation of the progeny, Z, W and Z, Z. This is how we'll get. So, this will be ZW will be the female here because females are heterogamous. Don't get confused. In birds, females are heterogamous. So, in humans, Drosophila, in grasshopper, insect and all that, that is some of the insects, the females are homogamous. In birds, the females are heterogamous. Always remember, females are heterogamous. Now, here there is one more mechanism, that is ZZ-ZO mechanism. In this mechanism, the female is again heterogametic, wherein they will not have a Z chromosome. It is called heterogametic, and male is a homogametic, that is ZZ. So, it is the same. So, here also it is, there is ZW mechanism, ZO mechanism. ZW in the case of birds, ZO in some of the other organisms. So, there is one more sex determination. So, in sex determination in humans, how it is done, we will look into. So, humans, we have 23 pairs of chromosome, wherein 22 pairs are autosomes. Like all of those are XX only. Okay, 22 are autosomes. One pair is a sex chromosome. That is, in female, we have 22 plus 1. That is 23 pairs of chromosomes we have, right? 46 chromosome. And in male also we have 22 plus 1. So, 22, all the 22 are homogamous, and this is also homogametic in female. In male, all the 22 are homogamous, only one is heterogamous. Understood? So, 23, 23. So, female, a pair of X chromosome XX, male, X and Y chromosome. Now, during spermatogenesis, the male produces two types of gametes, that is 50% with X chromosome and 50% with Y chromosome. Now, females produce only ovum with an X chromosome. So, here what did we have? So, we have the parents. This is the male. Male will have what? So, I have taken, they will produce what? X gametes with X chromosome and Y chromosome. Females will produce gametes with X chromosome and only X chromosome. Now, crossing over, what is it? So, this one we are crossing with this XX. So, therefore, it became a female XX. Now, this one again we are crossing with this. We get a female XX. Now, this we are crossing with this. We got XY. This again we are crossing with this. We got XY. So, if the child carries a Y chromosome, it will be a male. And male parent is only responsible whether the female will have a girl child or a boy child. Next, we'll look into sex determination honeybees. So, based on the number of chromosomes an individual receives, an offspring formed from the fusion of a sperm and the egg. So, sex determination in honeybees is decided based on these factors. So, what are the factors? Based on the number of chromosomes the individual receives, that is the progeny receives. And an offspring formed from the fusion of a sperm and an egg develops as a female queen or worker, and an unfertilized egg develops as a male drone by parthenogenesis. If you remember, do you know about parthenocarpy, wherein the formation of the fruit occurs without, or the seeds, the fruit formation occurs without the fertilization process, right? Same way here, those eggs which will get fertilized, it will develop into, that is, there is a male honeybee and there is a female honeybee. Now, fertilization will take place. Now, if fertilization takes place and then the eggs are produced, then the resulting progeny that is there, or resulting the baby honeybees that will develop, will either be a queen or it will be a worker. Okay. In that, the dominant one will be the queen and the recessive one will become the workers. Now, if there are unfertilized, now sometimes what happens, fertilization will not take place and even then the babies will develop. That is by a process called parthenogenesis, like how we have in fruits parthenocarpy. Here it is called parthenogenesis. Now, if such unfertilized eggs that are there, they will develop into the male drones. So, that is called as parthenogenesis. So, parthenogenesis is seen in honeybees. Always remember that. Males have half the number of chromosomes than that of the female. So, in honeybees, male will have half a number of chromosomes that of the female, means females will have more number of chromosomes. Male will have just half the number of chromosomes of the females. So, the females are diploid having 32 chromosomes. So, half. So, females how much they will have? They are diploid, meaning they will have 2n. They are 32 number of chromosomes. So, therefore, male are haploid having only half the set, 16 number of chromosomes. And this is called as haplodiploid sex determination system. So, what kind of sex determination do we have? We have XY sex determination system in humans. In honeybees, we have haplodiploid level of sex determination, because those honeybees which are having 32 number of chromosomes are all females. Those honeybees which are having 16 number of chromosomes are all males. This is called, what it is called, haplodiploid sex determination, because haploid is also there, diploids are also considered here. Males produce sperms by mitosis. So, mitosis means equational division. So, how much they'll have? Same 16 number of chromosomes will go to the progeny also. Males produce sperms by mitosis, hence do not have father and cannot have sons, but have a grandfather and can have grandsons. So, it's just that, so these males that are there, they usually produce sperms by mitosis. So, first actually mitosis happens and then the sperms get produced. So, if you see humans and all that, that is spermatogenesis process, if you see, first the spermatogonia will undergo mitosis and then they will undergo meiosis to produce the sperms. But here, up till the formation of the sperms, it is only mitosis that is taking place in the male honey bees. So this is about the sex determination in male honeybee. It is just very simple. So much is there. Always remember, if a honeybee is having 32 sets of chromosomes, then it is a female. If they have 16 sets of chromosomes, haploid chromosomes, then it is a male. So that is how it is. And now again in that we can separate it as drones, that is queen and workers. Queen and workers, how do we separate? So it depends on the dominance, and those which are having 16 set of chromosomes will all become that is drones without fertilization process, that is due by parthenogenesis, they become the drones. So this is about sex determination in honeybees. Next, talking about pleiotropy. So pleiotropy, as I said, a single gene which shows multiple phenotypic expressions. That is, one single gene will express multiple characters. Otherwise, usually what will happen, one single gene will represent only one character. That is, one gene is responsible for hair color, one gene is responsible for eye color, one gene is responsible for the color, that is, say for example, the texture of the hair. So it is like that, only one gene. But here, one gene shows multiple characters, is responsible for multiple characters. So the mechanism of pleiotropy is the effect of gene on metabolic pathways which contribute to different phenotypes. Say for example, phenylketonuria in humans. So there is a enzyme which is called phenylalanine hydroxylase. So this is the enzyme. So this phenylketonuria is actually a disease. It is a birth defect. So this birth defect usually occurs due to because of excessive accumulation of amino acid phenylalanine. So that amino acid phenylalanine should be hydrolyzed with the enzyme phenylalanine hydroxylase, so that phenylalanine doesn't get accumulated in the brain and doesn't cause brain damage and all that. But because of the mutation occurring in the gene, what will happen? That gene will not be able to produce this phenylalanine hydroxylase enzyme, which maintains the phenylalanine level in the body. Now, when this enzyme is not produced because of gene mutation, what happens? The same enzyme will cause a different character. That is, it will cause the accumulation of phenylketonuria, phenylalanine amino acid in the brain, leading to this particular disorder which is called as phenylketonuria. So, different characters they are controlling. One thing is, this gene is responsible for the production of this particular enzyme, and it is responsible for the maintenance of the amino acid, that is phenylalanine, all that. So, multiple characters it is doing, or multiple functions it is doing. So, that is called as pleiotropy. So, here actually a single gene mutation occurs. Which gene? The gene that is responsible for the production of the enzyme phenylalanine hydroxylase. So, this enzyme can control the production or accumulation of the phenylalanine amino acid. So, therefore, normal functioning of the humans can occur. So, if that doesn't happen, then it will result in the birth defect that causes some mental retardation and all that, because of accumulation of this phenylketonuria like happens. So, therefore, one gene controls several characters. So, that is called as pleiotropy. So, this is about pleiotropy. So, this was about this session wherein we learnt about the polygenic inheritance, taking the skin color of humans as example, and also we learnt about sex determination, that is how sex determination is done in humans, in Drosophila and humans, that is XY determination. In the case of birds, it is X, that is heterogamity. Birds exhibit heterogamity, females show heterogamity, therefore sex determination can be done. And also how sex determination is done in honeybees, wherein if 32 number of chromosomes are there, then they are females. If 16 are there, then they are males. So, we got to know. In the next coming session, we'll learn about pedigree analysis. In that, we'll learn about Mendelian genetic disorders and chromosomal genetic disorders. So, both of that we will learn. So, see you in the next coming session. Thank you.